Okay, for our next audio tour, we'll discuss the fibrous tunic in more detail. Uh, let's start at the limbus. It's a convenient landmark, both clinically and histologically. Dr. Murphy mentioned to you guys there's many different ways to refer to the limbus. There's the clinical limbus, which is the externally and clinically obvious junction of the clear cornea and the white sclera. And there's the histologic limbus, which is the wedge-shaped interdigitation of the knife-like sort of blades of cornea and sclera, where the posterior cornea extends further posterior. It's the sort of the inward sheath of the limbus, and the sclera extends axially, which is in a round structure, means sort of near the center. Towards the center, the outer sclera extends axially, and you get this overlap. But the white portion of the sclera begins about here clinically. And notice that the sclera is covered by bulbar conjunctiva, which we'll look at in a little more detail. So let's start with the layers of the cornea, remembering that that is the clear portion of the fibrous tunic. The number of layers varies somewhat from species to species. We'll show you there's an extra layer in the bird, as well as some other species we don't deal with too much, such as the elephant, but we'll get to that. So the major components of the cornea are the corneal epithelium, which remember is a stratified, non-keratinizing squamous epithelium. So stratified means it's many layers, squamous because they are squamous cells, but non-keratinizing because the layers maintain their nuclei all the way to the surface and don't undergo the process of cornification. We can see in this photomicrograph a thin band of the corneal epithelial basement membrane. It's not always very obvious, but remember that epithelial surfaces by and large have a basement membrane that they secrete on which they reside. We'll move inward to the corneal stroma, which, as you can see, makes up the bulk of the thickness of the cornea. It's composed of very regularly arranged collagen fibers, which are in turn composed of tiny fibrils that you cannot see with a light microscope. And there's a fairly regularly distributed population of spindle cells, which are keratocytes. Remember, kerato means cornea, so these are corneos. Essentially, that means corneocytes, or cells of the cornea. They're essentially a fibroblast. They secrete the collagen of the stroma, and although they look very quiescent and uninteresting, remember from Dr. Murphy's lectures that they actually have a very intricate network of processes that connect with each other. The analogy of everybody holding hands. So there's a lot of communication that goes on between these keratocytes in maintaining corneal health and clarity. We move inward to the glassy or hyaline basement membrane of the inner cornea, referred to as Descemet's membrane, or more accurately as the internal or posterior limiting lamina of the cornea. And that is a basement membrane which is secreted by the innermost layer of the cornea, the corneal endothelium. We'll zoom in as close as we can get to those guys so you can see they've got a lot of vacuoles, which is a characteristic feature. Remember that they're loaded with mitochondria. They pump a lot of ions out of the corneal stroma. Water follows passively, and these guys are primarily responsible for maintaining corneal dehydration, which is important for clarity. And remember that that is their major job, other than secreting Descemet's membrane. So if these are injured, whether it be by inflammation or increased intraocular pressure that makes them not work as well, their ability to remove fluid will be impaired, and you will get corneal edema. Just quickly, we're going to jump to the duct, and I'll just point out Bowman's layer, which was discussed by Dr. Murphy. We can see this brighter eosinophilic and acellular band, if you will, underneath the corneal epithelium. This is an area of modified stroma. The purpose of it is not known. And remember that the reason it's classified differently in some animals is because it's defined as something you can see at the light microscopic level. There may be something much thinner in dogs and cats, but we just can't see it, so they're considered not to have one. But you can find this in primates, humans, considering that we're primates, birds, elephants, but not in most of the domestic species. While we're on the bird, we'll just point out some of the modifications of the sclera. Here we can see a scleral ossicle, which gives rigidity to the sclera, offers protection, allows the eye to take on more complicated shapes or higher energy shapes than the general spherical shape of a mammal eye. And if you move posteriorly, we can see that much of the sclera is invested by scleral cartilage, which gives further rigidity to a larger portion of the sclera, and again facilitates protection of the globe and maintenance of abnormal or more complicated ocular shapes. And jumping back to the dog, again, the sclera is similar throughout. It's a little thicker at the equator. We can see the vortex veins, which become important in aqueous drainage. But I will point out one more time the lamina cribrosa as a posterior modification of the sclera, which is sieve-like and allows the bands of the axons to form the optic nerve to pass out of the sclera to bring the signals to the brain. So we see these thin bands of collagen which are running parallel to the sclera at this level. I think that concludes the fibrous tunic. I promised to talk to you guys about conjunctiva. So we'll zoom in on the conjunctival epithelium. So we can see that at the limbus, epithelium gets a little bit thicker and then it thins off, and we can see that it becomes fewer layers, and then as we move further back along the bulbar surface, it becomes sort of columnar, and if we're lucky, we can find a goblet cell. The dog is not cooperating, so we'll jump to the cat. See if we've got any goblet cells in our cat. Perfect. This cat has a little bit of keratitis and conjunctivitis, but here we can see the single or double layer of cuboidal to columnar epithelium and these big, conveniently goblet-shaped cells with the the granular basophilic material, which is mucus, is secreted from these, and that contributes to the mucus portion of the tear film, which was covered by Dr. Murphy. Okay, that wraps up this portion. We will move on to the vascular tunic in the next installment.